All right, let's look at carbohydrates. So carbohydrates are molecules made up of carbon, hydrogen, and oxygen. Those three are present. Typically, they form a ring. This is a 1, 2, 3, 4, 5, 6, if you count this carbon up here, ring with an oxygen in the ring itself. Or you could have in fructose, for example, you have a 5-carbon ring, and oxygen's present in the ring there. So this is a monosaccharide, mono meaning one, saccharide meaning sugar. This is a disaccharide, di meaning two, again saccharide is sugar, and a polysaccharide. Poly means many and saccharide is sugar. Oxygen and hydrogen and carbon, it says, are always in a ratio of 1 to 1. So C6H12O6, as in glucose here, there's 6 carbons, 12 hydrogens, and 6 oxygen. It's 1 to 1 for carbon, hydrogen, and oxygen. Let's go up. So carbohydrates are linked through dehydration reactions, just like anything else. Mr. Kincaid, I'm trying to record this video for the 5th time. Will you please be quiet? I'm sorry. I appreciate that, but that's going to be on YouTube because I'm not doing it again. So
I'm sorry.
I just want to throw my iPad out the window.
I'm sorry.
Many monosaccharides such as glucose and fructose are reducing sugars, which means that they will react with Benedict's reagent. And the part that they react with is the copper, or the cupric part of it. I'm not going to talk about exactly how that happens, but just know that if it's a single monosaccharide, a single sugar, that it will react with the Benedict's solution. If you have just a few of them, it will turn green, but if you have a whole bunch of them, it will turn reddish orange. And if it's not present, if you have sucrose, which is like a polysaccharide, it will turn or stay blue. So Benedict's solution will not change if you have a polysaccharide. So here's how you're going to do it. You're going to get 7 test tubes and you're going to number them 1 to 7. And you're going to add the materials from table 1 that's in the back of your page of your packet. You'll have onion solution and onion juice, I think is what it's called, potato juice, glucose solution, sucrose solution, starch solution. So you're going to add those to your tubes, and then you will add 2 milliliters of Benedict's solution to each tube, and you will mix. And then you will place the tubes in a boiling water bath for 3 minutes and observe the color changes during the time that it happens. After the 3 minutes, you'll remove the tubes and let them cool to room temperature, and then you'll record the color of each test tube in table 1. So those are for monosaccharides like this. Sucrose, I'm sorry, I said it's a polysaccharide. It's actually a disaccharide. Sucrose should not react in that test. But you should be able to identify starches, which are polysaccharides, but they are coiled polysaccharides. And you can test them by staining them with iodine. So it says it distinguishes starch from monosaccharides, disaccharides, and other polysaccharides because starch is a coiled polymer of glucose. It's a bunch of glucoses stuck together. Your plants actually make starch. That's how they store their sugar. Iodine interacts with these coiled molecules and becomes bluish black. So if you remember from our osmosis diffusion lab back in regular biology, you had your iodine in the beaker and you had your starch in your little dialysis tube, and when the iodine diffused into the tube, it turned the tube blue. So that's the color change that we're going to look at. If you have a lot of starch present, it will turn dark purple. If you have glycogen, which is found in animals, but it's slightly different than the starch in plants, it will kind of be a pinkish or an intermediate color. So you're going to use the same 7 test tubes as before. You'll just rinse them out and reuse them. You'll add your materials as shown in table 1, and it's going to be the same stuff as you used before. And then you'll add 3 to 5 drops of iodine, and you'll mix and record the color of the tubes. Pretty straightforward for that one. There's no water bath involved. Next, we'll talk about proteins.